population genetics, I looked up two definitions, one in the Stanford and Wikipedia, which is fairly generous. It's the on the genetic composition of biological populations and so on. Whereas if you go to Wikipedia, it becomes a lot more quantitative, and I almost think that this definition was written for by someone from the HLA field, because it's what you can read here is population genetics is the study of the distributions and changes of allele frequency and so on. So it's on frequencies of alleles, and as you will see also on haplotypes, and how they are created and changed by the evolutionary process. I'm not talking about that, but I just want to draw your attention to the fact that genetic-driven mutation gene flow, in particular the population structure, is something that we are still struggling with in the HLA field in the registry context. So what I would like to talk about, and I wonder how long I still have. It was
it doesn't matter.
It doesn't matter?
No, it doesn't matter. No,
okay. Just wondering if the lunch is walking away.
No, I answered that you're going to be later on.
Okay, good. So I will go briefly into the basics of HLA polymorphism and frequency estimation, and then focus on donor registries and typing strategies in donor search. I will not go into what Frida mentioned, the optimal number of donors. This will be a completely separate talk. But of course, it's a very, very important question for people thinking about creating a registry. I suppose that you are familiar with the short arm of chromosome 6 and the important loci of HLA. This is one of the slides I have hated for a long time because I had to update it every other week. I hope it works. I have stopped updating it a couple of years ago because it does not matter if you see 1,000 or 2,000. It just tells you that first, the number of alleles, the number of variants that we have to deal with in HLA is huge, and the increase is still tremendous. And if you translate that into real numbers of genotypes at one locus, haplotypes, possible haplotypes, and possible phenotypes, you're ending up in numbers which are well beyond the population of the Earth. So you have HLA combinations which are frequent and those which are rare, and the vast majority of HLA combinations which are theoretically possible do not exist on the globe. However, they come into the problem that we are dealing with at the maximum likelihood estimation of HLA frequencies. You usually have HLA data which are phenotypes, and you don't know the haplotypes. And the frequency of the haplotypes is what is relevant for many things that you are doing in donor search and registry planning and so on. And this maximum likelihood estimation wants to determine the frequencies, the values of what we call a parameter vector of haplotype frequencies. And the length of the complexity of this vector depends on the number of haplotypes that might be present in a sample. And I don't want to go into the math. This is just to scare you off a little. The underlying principle is that you can estimate haplotype frequencies even if you don't see them, if you are unable to observe them in the sample that you have. This is roughly a situation like in CAT scan, when you have a single planar picture of a three-dimensional body. You never can impose what you are seeing. But if you have several of those and if you apply certain principles, you can observe by trivial linear or planar pictures, you can infer a three-dimensional structure. This is exactly or very similar to the complex mathematical EM process, this maximum likelihood process, which brings you back from the phenotype frequencies to the unobservable haplotypes. On the other hand, if you know haplotype frequencies, inferring, calculating the frequency of a phenotype, you can do it with a pocket calculator. That's easy. What brings those two calculations together is the Hardy-Weinberg principle, which is the underlying math. And I don't want to give a class in biostatistics, but you should always be aware that in most cases you not only have to multiply the frequencies of the two alleles or two haplotypes, but as long as they are not identical, you also have to apply another factor of 2. The EM algorithm for the frequency estimation is basically easy to implement, but the complexity comes not primarily from the number of individuals, which can go into the millions in registries. It increases a little bit the number of loci considered. So you start with 2 and can go up to 5 or 6 in stem cell transplantation. The real nuisance starts with the typing ambiguities. If you have those NMDB, multiple allele codes, generic typings in there, and it really gets bad when you want to include loci like HLA-C or DQB1 in your estimation, and you have individuals in your sample which aren't even typed for those loci. There are a couple of other things which made me advise you, if you consider doing that, and you have a sample which is not completely high resolution at all loci you consider, you will fail with those tools that are readily accessible on the internet, like Arlequin or a couple of others. I will show you a number of pictures and a number of applications that you can do when you know haplotype frequencies. And this is from a talk I gave about 10 years ago in Tokyo, and we started using data from bone marrow donors worldwide to estimate 3 locus ADDR serological level haplotype frequencies from the global population. This was roughly the consensus resolution that was accessible at that time. And to explain this type of graph, the x-axis means the size of the registry, and the y-axis is the percentage of patients which would find a donor in a registry of that size. And you have now here 6 populations, the top 3 lines, and you can read that. They are all from the British Island, from the UK. And why are there 3? Because for the UK alone. The top one is from the Welsh registry. The second one, the second from the top, is from the British bone marrow donor and platelet panel. And the third one is from the Anthony Nolan. And why do they look different? It's very simple. The more homogeneous the population, the higher up the curve will be, because you will find more donors for your own patient given the same registry size. And Wales is homogeneous. And the British panel is mainly recruiting outside the London area, which is extremely heterogeneous. So Anthony Nolan is a lot more polymorphic than the others. And then if you go down, it's Germany, Spain, and Italy. Italy is the most polymorphic population of those 6 in Europe. And these are other European populations. The top 2 are Ireland and Finland. Ireland is a geographical island, and Finland is a language island. So they make a very homogeneous population, and this is also what could be a consolation for the Armenians, which are very special, but it does not really impair the chance of finding donors, because if you have a homogeneous gene pool, your situation is a lot easier. However, what you can do is not only look at a registry for their own country. This is a similar graphic looking at the situation of German patients with donors in other countries. And essentially all the curves which are close to the black German curve are European populations, and this is why we have done a pretty good job in the transplant medicine while not having a real registry in the late 80s. And if you look at those 4 curves down here, they are Japan, Hong Kong, Taiwan, and Thailand. We will hardly ever find a donor for a German patient in those registries. And now to switch this view to the top down, this is the curve for Japanese patients with Japanese donors. It's the highest in the world. They are an island, and they are so extremely homogeneous. And even their closest neighbors here, Hong Kong, Taiwan, and Thailand, are far away. I now want to take a different point of view. The graphs I've shown you so far were on different populations. The next few graphs I'm going to show you are on the German population, but analyzed at different typing resolutions. The top line here is roughly where we came from. It's A, B, DRB1, generic resolution DNA typing. So at about 1 million donors, you are finding a match at lower resolution 3 locus level for about 87% of the German patients. However, only for about, let's say, 57% if you're going, yes, 57, if you're going at 5 loci, ABC, DRB1, DQB1 at high resolution. But what I'm going to show you is how it looks in between. If you're not able to afford 5 locus high resolution typing immediately. So if you're only doing high resolution for 3 or 4 loci, this is how it looks. 3 loci, this curve. If you add HLA, DQB1, it doesn't bring a lot of information. However, if you add HLA-C and just leave out DQ, it's almost as good as 5 locus matching. So you can, I'm adding 2 more curves, but the main message I will want to bring to you is on the following slide. This shows you what the probability of 5 locus high resolution matching is if you have a match at the level depicted on the left side. So if you only have A and B generic, 4% of the AB low resolution matches are really ABCDR&Q high resolution matches. The next most important point of information is adding DRB1, and then it's adding C. It's not going to high resolution anywhere. HLA-C is reflecting already at low resolution a large part of the polymorphism. And what you can see here is if you have 4 loci at low resolution, you already have paid 50% of the rent. And then to go to high for DRB1 and then for HLA-B, and then you're already pretty close. This would be a typing strategy if you, for example, assume that high-res typing is twice as expensive as low-res and all loci are equal. This may be obsolete already when you go to NGS and everything's cheap and you're doing everything at once in full high-res beauty at 5 loci or even more. Okay, better hurry up. Just a few words. Which donors should be better typed? Of course, younger donors, they stay longer in the registry and they are selected more often. And evidence has increased that an age difference of about 25 years is about as detrimental for overall survival as a single mismatch. Male donors are selected more possibility, but evidence is not as good. And of course, you could try to cherry pick interesting HLA, but this has only a limited efficiency except for the very beginning, and I would like to discourage you to go for the rare HLA types. Rare HLA types are rare also in patients and are of little use in the end. So if you currently the HLA effects are much less important than other attributes of the donors. There are a number of curves here, but only look at the 3 through lines. This is the lowest one is 5 locus high resolution matching for A, C, P, DRB1, DQB1. The next curve, now is again a different point of view, is the procurement rate of single mismatched donors, and the top one is of 2 mismatched donors. So if one is able to accept a single mismatch, this is really saving you a lot of, or covering up for a lot of this insufficiency, because even going to 100 million donors will not procure donors for over 90% of your patients. And this is a theoretical thought. So my message is here, donor recruitment will not solve the problem of all patients as long as we cannot accept a single mismatch. This is how a typical match list looks. It's a screenshot taken from the BBW website from one of our patients. And it proves that searching for a matching donors is often like searching for the needle in the haystack. This was an event we made where people had to look for a color pattern that we had fixed to the tips of the needles in the haystack. So it was a sort of a lottery. Where are the challenges coming from? And you have seen this from in the BBW screenshot. The HLA data are so heterogeneous because they have been acquired over a long time with varying typing methods, even mixing serology and DNA. You're getting broad assignments, multiple allele codes that nobody can memorize. They have grown to 5 letter codes in the meantime. And you often are missing data for relevant loci. And there are some what I call inborn errors of the HLA nomenclature. I'm not sure if you can really see that. This is an example of a match list where you see the HLA of the patient up here, and then the HLA A, B, C, TRB1, DQB1. And this is how a match list typically looks, and it's even not a bad one because it's already color coding mismatches here and there, but I have a difficulty even pinpointing it at this angle, and allelic difference are shown in blue and antigen difference are shown in red. But due to these, and I will go through it a little bit faster in the interest of time, if you look at the ambiguity boiled down to the protein sequence of the antigen recognition site, even the patient has 4 possibilities. The first donor has about over 200,000 possibilities, and it goes up to 5 times 10 to the 17th for the last donors with AB serology, no HLA-C and DQB1. And what makes things easy for us, and what is the real point of the whole thing, is that although we have about 1 trillion haplotypes, which are theoretically possible, only about 100,000 show up in the German donor population. So this allows us to boil down this myriad of possibilities, and I will skip a few slides, bringing these huge numbers between 200,000 and, between 200,000 and 10 to the 17th to a fairly handleable amount between 2 and 200 possibilities. And then you can really add what we want to see, the probabilities. 10 out of 10, going down from 97% in the first case to 56 for the third donor. And it shows you the probability of only being 9 out of 10 and being matched or mismatched at each of the loci. I'm skipping the validation of this whole OptiMatch approach and just tell you, if you have such a tool, you are a lot stronger searching for the best match for your patient. A few examples from the application of the haplotype frequency-based matching prognosis. Here you see that in this match list, if you can't get one of the first donors, a missing C is irrelevant because the match probability is only going down to about 80%. It's a lot worse if you have C, but you're losing the high resolution on the DR and PQ, which is highly selective. And even a lot worse when you're, in this case, when you are lacking the DQ, which is rare because DQ usually is not highly selective. And in another case, you see that the best donor is even lacking C and DQ because the high resolution on the 3 other loci is sufficient. And if the resolution goes down, the probability goes down a little. And if you have another donor which has been typed for all 5 loci, but only at low resolution, the probability goes down to 28%. It's not really helpful anymore. And it's not, neither one, going for all loci or going for high resolution is intuitively clear which one is the more favorable. It really depends on the patient's situation. And you see then, missing C and DQ does not further impair the situation. It only goes from 28 to 24%, and so on. One last point is if you have a number of potential 10 out of 10 matches, and the program tells you that they most likely will turn out not to only have a single, but even more than a single mismatch, because the probability for 10 out of 10 is negligible, and even for 9 out of 10 is well under 10%. You should resort early to a safe 9 out of 10 donor, because waiting and searching too long is more detrimental for the patient than a single mismatch. So, what I have shown you briefly is that the known haplotypes can reduce the ambiguity on donor list by many orders of magnitudes. And you can use haplotype frequencies, not manually, but in the computer system, to calculate matching probabilities for ambiguous loci. Our goal is really to put you the needle from the haystack onto a silver tray. And this is the end of my talk. I want to thank the volunteer donors in the world, which are the basis of our work, all our colleagues and partners within Germany and around the globe, and the people who have done the work that I presented here, mainly the OptiMatch team. You're seeing the names, and in particular I want to thank Professor Marsh in London, who is maintaining the HLA nomenclature and is herding all our HLA cats on the globe. So this is my staff in Ulm, and kind regards from our city, and I hope we will have a better return home.